Dear student, I am Dr. Prakash Mungli. In this glycogen metabolism lecture, I will be explaining you on regulation of glycogen metabolism. Now let's get into the details. Glycogen metabolism, a regulation, it can be done through hormonally or allosterically. Now the regulation of glycogen metabolism occurs in different ways. It can be hormonal regulation or it can be allosteric regulation. And also regulation we can study that happens in different tissues. That can be in the liver or in the skeletal muscle, because liver and skeletal muscle significantly contribute to the amount of glycogen that is present in our body, and also especially liver. The purpose of glycogen that is present in the liver is for maintaining the blood glucose. So now let's get into what are the regulatory points related with glycogen metabolism. Now the liver glycogen is regulated based on the insulin-glucagon ratio, and we consider fasting state and fed state to understand the effect of insulin and glucagon ratio on glycogen metabolism in the liver. And also glycogen metabolism in the liver can also be regulated by the presence of epinephrine. And you know, epinephrine will be released in our body in response to hypoglycemia, in response to exercise, and in response to stress. Now, while the regulation of glycogen metabolism in the skeletal muscle, as I said before, it can again be because of hormonal regulation or allosteric way of regulating glycogen there. One is the AMP levels in the skeletal muscle can affect glycogen metabolism. Other is the calcium levels in the skeletal muscle can affect glycogen metabolism. And also the effect of epinephrine on the skeletal muscle, especially when person is exercising or in stressful situation. Only thing that you must be aware and be careful with the glycogen metabolism in the skeletal muscle is there is no effect of fasting on skeletal muscle glycogen stores. No effect of fasting on the skeletal muscle glycogen stores. It is because glucagon receptors are not present over skeletal muscle. The fasting hormone glucagon do not have any effect on the glycogen stores in the skeletal muscle. Now the two enzymes that are regulated in the glycogen metabolism regulation that you must be aware of is glycogen synthase, which is the key regulatory enzyme in glycogen synthesis, and glycogen phosphorylase, which is a key regulatory enzyme in glycogen degradation. These two enzymes are regulated both allosterically to meet the needs of a particular tissue, or hormonally to meet the needs of our body as a whole. Let's get into more details on how glycogen metabolism is regulated both in the liver and in the skeletal muscle, hormonally and allosterically. Now first is allosteric regulation of glycogen in the liver. So what we are going to do now is we are going to separate this regulation as the kind of regulation that will be going on under well-fed condition. That means after a food, what kind of regulation will be going on in the liver. And also we are going to consider the regulation that will be going on under fasting condition. Now again we will be taking two enzymes to understand this regulatory mechanism. One is the glycogen synthase, other is glycogen phosphorylase. Now let's see what kind of allosteric regulation will be going on in the liver under fed condition, especially on glycogen synthase and glycogen phosphorylase, and then we will move on to the fasting state. Now the glycogen synthase under fed condition, it will be positively modulated by glucose 6-phosphate. The reason for that is, as we know, when we are in fed condition, so glucose, which is at high concentration, in the blood, entering into the liver, and glucose getting into very first reaction there, converted into glucose 6-phosphate. And the glucose 6-phosphate, although it is going into glycolysis, but some of the glucose 6-phosphate is diverted into glucose 1-phosphate, and this glucose 1-phosphate can be converted into UDP glucose, and that UDP glucose is going into glycogen formation. This is what is the glycogen synthesis that we have seen in our previous lecture. Now note that in the glycolysis, when glucose 6-phosphate is going into fructose 6-phosphate, and fructose 6-phosphate goes into fructose 1,6-BP, and now that fructose 1,6-BP, it is catalyzed by an enzyme called PFK1, that is phosphofructokinase 1. Now this phosphofructokinase 1 is the most regulated enzyme in the glycolysis, especially when the person is in fed condition. There will be a lot of ATPs and citrate in the cytoplasm, and also there will be more fructose 2,6-bisphosphate. So during this time what will happen? So because of the presence of higher levels of ATP and citrate, so these two molecules will have a negative effect on PFK1. And also when excess glucose is there, entering into the liver, converting into glucose 6-phosphate by glucokinase enzyme or hexokinase in other tissues. So more and more glucose 6-phosphate is flooding into glycolysis, so that might saturate a PFK1 enzyme itself. PFK1 enzyme can reach its Vmax, maximum velocity. At that time what will happen? So there will be buildup of fructose 6-phosphate, which will be converted back into glucose 6-phosphate because it's a reversible reaction. And whenever glucose 6-phosphate is kind of building up, at that time that glucose 6-phosphate will have a positive effect on glycogen synthase enzyme, and here in the pathway glycogen synthase comes here. UDP glucose into glycogen done by glycogen synthase enzyme, so that is positively modulated by glucose 6-phosphate. Same thing happens even in the skeletal muscle also. So this is how glucose 6-phosphate, whenever it is kind of building up in the cytoplasm of the liver, skeletal muscle or other tissues, it will have a positive effect on glycogen synthase. Now when the glycogen synthesis is going on, at the same time glycogen degradation should not go on because it's an antagonistic pathway, right? So same glucose 6-phosphate will have a negative effect on glycogen phosphorylase enzyme. Okay, let me just show you that here, just connecting the pathway. So glycogen degradation into glucose 1-phosphate under fasting condition done by glycogen phosphorylase. That's the main enzyme there. And the glycogen phosphorylase, it is negatively modulated by glucose 6-phosphate. Now that makes sense here, right? You can't run two opposite pathways at the same time. And also, what are the other negative modulators of glycogen phosphorylase? It is ATP. Whenever ATP levels are plenty in the liver, there is no need to break glycogen, because what is the purpose of breaking glycogen? Just to provide glucose to the blood by the liver or in the skeletal muscle, just to make more ATPs. When ATPs are already there, there is no need to break glycogen. That's why ATP act as a negative modulator on glycogen phosphorylase. Similarly, when we have plenty of glucose also has got a negative effect on glycogen phosphorylase. Now coming to the fasting condition. Under fasting condition, what will happen? There will be lower glucose 6-phosphate because under fasting condition there is a need for glucose in the blood. So glucose 6-phosphate levels are lower, glucose levels are lower, and ATP levels are lower. That's why glucose 6-phosphate do not have a positive effect on glycogen synthase. So glycogen synthesis don't go on. And the glucose ATP levels are, since they are lower, so they don't have a negative effect on glycogen phosphorylase. That's why glycogen phosphorylase is now free to work. And this is how regulation is going on in the liver, especially allosterically. You can see that here in the pictorial form. As you see, the points that I have explained in the previous slide, you see the glucose 6-phosphate is a positive modulator on glycogen synthase enzyme, whereas same glucose 6-phosphate is having a negative effect on glycogen phosphorylase. Because of this, glycogen synthesis will increase, whereas glycogen degradation will decrease, because both are antagonistic pathways. At the same time, presence of higher levels of ATPs, presence of higher levels of glucose in the liver will have a negative effect on glycogen phosphorylase, thereby glycogen degradation will decrease. Now let's move on to see allosteric regulation of glycogen metabolism in the skeletal muscle. Now the similar points that I have explained in the previous slide applies here. Glycogen synthase under well-fed condition is positively modulated by glucose 6-phosphate. Same glucose 6-phosphate will have a negative effect on glycogen phosphorylase. ATP glucose will have a negative effect on glycogen phosphorylase. So all those points are similar what we have seen with the liver also. Now during exercise what happens? Because as I already told you, there is no effect of fasting on the skeletal muscle glycogen metabolism. So that's why it is not written here. So what is written is exercise. During exercise what happens? During exercise, as we can expect, that the glycogen stores that are present in the liver need to be broken down, because there is a need of energy there, right? So whenever a skeletal muscle is contracting, so as you all know, ATP breaks into ADP, and ADP further breaks into AMP, and AMP levels will rise, and this rise in the AMP and will have a positive effect on glycogen phosphorylase enzyme and keep the enzyme active, because AMP is a energy hunger signal for the cell. That means the stores, glycogen stores, which has the energy there, means in the form of glucose, it has to be broken down. So glycogen has to break into glucose 1-phosphate, so that glucose 1-phosphate goes into glycolysis. So that is positively modulated by AMP. So AMP will keep glycogen phosphorylase active, thereby glycogen breaks into glucose 1-phosphate, glucose 1-phosphate goes into glucose 6-phosphate, and glucose 6-phosphate into fructose 6-phosphate and fructose 1,6-bisphosphate in the glycolysis. And also note that the PFK1, the rate limiting enzyme in glycolysis, PFK1, that is also positively modulated by AMP. So note how this AMP is making sure skeletal muscles are getting sufficient energy whenever they are when skeletal muscle is contracting. First thing is it is breaking glycogen by positively modulating glycogen phosphorylase using AMP, and then further down in glycolysis, same AMP will have a positive effect on phosphofructokinase 1 enzyme. Remember that point. Now let's look at the figure. So understand it much better using the figure, and also kind of revision with what I have explained you in the previous slide. Now the glycogen synthase positively modulated by glucose 6-phosphate. Same glucose 6-phosphate will have a negative effect on glycogen phosphorylase. So antagonistic pathways are simultaneously controlled, and the ATP will have a negative effect on glycogen phosphorylase in the skeletal muscle. Plenty of ATP means why to break glycogen, right? So that's why ATP will have a negative effect on glycogen phosphorylase. Now AMP, now think in the opposite way. When you don't have plenty of ATPs, that means you will have more AMPs in the skeletal muscle. So AMP will activate glycogen phosphorylase and release glucose 1-phosphate. What I was saying was, this glucose 1-phosphate will be converted into glucose 6-phosphate, which is going on in the skeletal muscle. I am just writing it outside because there is no much space here. Now the glucose 6-phosphate converted into fructose 6-phosphate, and fructose 6-phosphate goes into fructose 1,6-bisphosphate. Okay, so this is catalyzed by a enzyme PFK1 enzyme, and that's an irreversible reaction. Otherwise, other reactions here, they are all reversible. Now this PFK1 is the master controller of glycolysis. So after that, like fructose 1,6-bisphosphate goes all the way to say pyruvate, and pyruvate has a different fate. It can go into TCA cycle or it can go into anaerobic glycolysis. All those fates are there. Now AMP, the same AMP which was positively modulating, and the glucose 1-phosphate going into glycolysis. So same AMP will have a positive effect on PFK1, thereby glycolysis increases. Ultimately pyruvate is made, and pyruvate going into TCA cycle, so gives NADH and FADH, or pyruvate going into lactate give ATPs at the substrate level phosphorylation. So like this, ATPs can be synthesized whenever skeletal muscle is contracting by breaking the stored energy as glycogen. Now let's understand hormonal regulation of glycogen metabolism, especially in the liver. So in this particular figure, I am taking example of glucagon and epinephrine, which is binding to beta receptor. Now that means I am going to explain you the glycogen metabolism regulation under fasting condition only, and later I will touch upon what happens under fed condition. This particular figure predominantly showing the glycogen metabolism that is going on under fasting condition. Alright, so let's begin from glucagon and epinephrine binding to glucagon and epinephrine receptor. When glucagon binds to glucagon receptor, epinephrine binds to epinephrine receptor. So the receptor gets activated, and note that glucagon receptor and epinephrine receptor, which is a beta receptor here, these are G protein coupled receptor, and these are a serpentine receptor. Serpentine receptors. Serpentine means looks like a snake, and as you see here in the figure, it looks like a snake, right? So this serpentine receptor, which is G protein coupled receptor, so it has G protein, as you know, it has alpha, beta and gamma subunit. And now because of the binding of either glucagon binding to glucagon receptor, epinephrine binding to epinephrine receptor separately, so alpha subunit in the G protein is separated from beta gamma. Now the alpha subunit is, this happens whenever GDP is replaced by GTP. Now the GTP bound alpha subunit, it is going to activate a membrane bound enzyme called adenylate cyclase. And what this adenylate cyclase does, adenylate cyclase is going to break ATP into cyclic AMP. Now so that will rise the cyclic AMP levels. When the cyclic AMP level rises, so it is going to activate protein kinase A enzyme by binding to its regulatory subunit. Cyclic AMP will go and bind to regulatory subunit of protein kinase A. Thereby, cyclic AMP is going to activate a protein kinase A enzyme. Now, the active protein kinase A, what it does, it is ultimately, the idea here is to ultimately activate glycogen phosphorylase enzyme, because we are like the hormones that are binding here is glucagon and epinephrine, and you know glucagon and epinephrine, they are trying to increase the blood glucose level under fasting state or stressful situation, right? So ultimately the idea here is to increase glycogen phosphorylase activity and decrease glycogen synthase activity, right? So in order to achieve that, what protein kinase does is, protein kinase A, it is going to phosphorylate initially an enzyme called glycogen phosphorylase kinase. Now this glycogen phosphorylase kinase, as it is there in the name, once it is phosphorylated, it becomes active and it will go and activate a glycogen phosphorylase enzyme, because glycogen phosphorylase is active only when it is in phosphorylated state. So the glycogen phosphorylase kinase is going to add phosphate to glycogen phosphorylase and keep it active. So it's a kind of two-step process. First, protein kinase is activated, and that protein kinase is going to activate glycogen phosphorylase kinase, and that glycogen phosphorylase kinase is going to activate glycogen phosphorylase. And once glycogen phosphorylase is active, as it is shown here, glycogen breaks into glucose 1-phosphate by adding inorganic phosphate, and glucose 1-phosphate converted to glucose 6-phosphate, and glucose 6-phosphate is converted to glucose by glucose 6-phosphatase enzyme, and that gets into the blood, maintain the blood glucose level. Now when the glycogen degradation is going on one side, the antagonistic pathway, that is the opposite pathway, glycogen synthesis, should not go on. So the same protein kinase A, it is going to go and add phosphate to glycogen synthase, and the phosphorylated form of glycogen synthase is an inactive form of an enzyme. That's why glycogen synthesis will decrease. So this is how glucagon and epinephrine will make sure glycogen degradation is going on under fasting condition and stressful situation in the liver, glycogen synthesis goes down. Okay. Now I want to explain you one more point which is kind of related to this particular metabolism here, regulation, and that is, as you see here, the cyclic AMP, it will be converted into just AMP. Cyclic AMP is a second messenger, AMP is not a second messenger. So that cyclical process is taken out here, right? So the enzyme that does is a cyclic AMP phosphodiesterase enzyme. Now the cyclic AMP phosphodiesterase enzyme, it is controlling the levels of cyclic AMP in the cell, thereby this cell signaling process and the metabolic regulations are kept under control. Now there are certain things that can inhibit the cyclic AMP phosphodiesterase enzyme, and that is caffeine in coffee, and theophylline in tea, and then we have theobromine in cocoa. So all of that will have a negative effect on cyclic AMP phosphodiesterase enzyme. Thereby cyclic AMP phosphodiesterase activity decreases because these molecules will have a negative effect. So when the cyclic AMP phosphodiesterase enzyme activity goes down, that means cyclic AMP levels are kind of accumulating, building up, because it is not degraded into AMP. So when cyclic AMP levels are more, so glycogen degradation is going on at a higher rate, because higher cyclic AMP means higher activity of protein kinase A, that means higher degradation of glycogen and less glycogen synthesis. This is what happens when we drink tea, coffee or cocoa, right? Anything that has been added with cocoa. So because they have caffeine, theophylline, theobromine, they all will inhibit cyclic AMP phosphodiesterase, thereby increases glycogen degradation. And this is the reason why in a long distance run or anything, any sporting event which needs a long endurance, so something called as carbohydrate loading is done 3 to 4 days prior to the event. So at least like 4 days prior to the event, a person who is participating in the event will exercise to the maximum, and so that glycogen degradation is completely done. I mean, almost entire glycogen in the skeletal muscle is kind of degraded, and then the person takes rest for 3 days and eat lot of carbohydrate food, like high carbohydrate food. And during this time, these 3 days of rest, well-fed condition, there will be more and more glycogen synthesis going on, replenishing all the glycogen stores. Like glycogen stores are filled in the skeletal muscle, full. And before 30 minutes before the activity begins, a strong black coffee is consumed which contains caffeine, and that will keep cyclic AMP phosphodiesterase inactive and thereby maintain high levels of cyclic AMP, and that will keep glycogen degradation efficiently going on. And this is exactly why a carbohydrate loading is done prior to the sporting event. So I hope you understood this concept and the application there. Now let me briefly explain you what will happen when a person goes into fed condition. When a person goes into fed condition, now it's all about insulin. Glucagon will decrease. Person, if it is restful, epinephrine decreases. Now insulin increases. Now what this insulin does, as it is shown. So now in the fed condition, glucose levels will increase, and that glucose will get down into the hepatocyte. So now all the dotted arrows that are shown here, they will become a bold arrow. Okay, so glucose will be entering into the liver, and glucose converted to glucose 6-phosphate by glucokinase enzyme, glucose 6-phosphate into glucose 1-phosphate, glucose 1-phosphate converted to UDP glucose. Now you have UDP glucose there. And insulin has to keep this glycogen synthase active, because previously glucagon and epinephrine has kept this glycogen synthase inactive by phosphorylating it. Now that phosphate has to be removed from the surface of glycogen synthase. So what insulin does is, insulin always activate an enzyme called protein phosphatase 1. And that protein phosphatase is going to remove phosphate from the surface of enzymes. That includes glycogen synthase. So bring that back into active condition, and that's why glucose can go into glycogen synthesis. So glycogen synthesis increases. When the glycogen synthesis is increasing, at the same time glycogen degradation has to decrease. And how that is done? By insulin. Same protein phosphatase, which is activated by insulin, it is going to remove phosphates from the surface of glycogen phosphorylase enzyme, which is an active enzyme, and keep it inactive. At the same time, same protein phosphatase enzyme, it's going to remove phosphate from the surface of glycogen phosphorylase kinase and remove phosphate and keep it inactive. So thereby glycogen degradation is kept low, glycogen synthesis is kept high. And this is exactly how glycogen metabolism is regulated by glucagon, epinephrine under fasting and stressful situation or exercise, and insulin under fed condition. Now let's move on to see what is the effect of epinephrine when it binds to alpha receptor. Now note that in the previous slide I have explained the effect of epinephrine binding to the beta receptor. Now in this slide I am going to explain you the effect of epinephrine binding to the alpha receptor. Okay, so the epinephrine, when it binds to alpha receptor instead of beta receptor in the liver, this alpha receptor again, it's a G protein coupled receptor. Only thing is, here there will be activation of phospholipase C enzyme, and this phospholipase C enzyme is going to break PIP2 membrane phospholipid, that is phosphatidyl inositol 4,5 bisphosphate. What I am saying here is PIP2 is phosphatidyl inositol bisphosphate. Phosphatidyl inositol bisphosphate is a membrane phospholipid. So phospholipase C enzyme is going to break this phosphatidyl inositol 4,5 bisphosphate. So the enzyme is phospholipase C, PLC, phospholipase C. It's going to break this molecule into IP3 and DAG. IP3 is inositol triphosphate. Inositol triphosphate is IP3, and DAG is, so that is plus, DAG, diacylglycerol, that is DAG, diacylglycerol. Diacylglycerol means glycerol containing attached to the 2 fatty acid. Okay, so that's what phospholipase C do whenever it is activated when epinephrine binding to alpha receptor. So we have two molecules coming out of PIP2, IP3 and DAG. What is the role of IP3 and DAG? IP3 will go to endoplasmic reticulum and release calcium from the endoplasmic reticulum. the calcium in the cytoplasm will bind with a calcium binding protein called calmodulin. So the calcium calmodulin is going to activate calmodulin dependent protein kinase, and it's going to activate a glycogen phosphorylase kinase enzyme. Now this calmodulin dependent protein kinase, it is going to phosphorylate glycogen synthase, and phosphorylated form of glycogen synthase, it's an inactive form. Thereby it decreases glycogen synthesis. And also since glycogen phosphorylase kinase is activated, so it is this glycogen phosphorylase kinase, it is going to phosphorylate glycogen phosphorylase enzyme, thereby keep it active. So even the protein kinase C, which is being activated on the other side by diacylglycerol activating protein kinase C enzyme. So that protein kinase C is going to keep glycogen synthase inactive and glycogen phosphorylase active. So overall, glycogen phosphorylase is activated, and once the glycogen phosphorylase is activated, it's going to break glycogen into glucose 1-phosphate and then into glucose 6-phosphate forms, and then that forms glucose. And that is how epinephrine binding to alpha receptor will work in the liver. Now let's move on to see how glycogen metabolism is regulated in the skeletal muscle. When a person is exercising, in response to exercise, there will be release of epinephrine. And the epinephrine binding to beta receptor over skeletal muscle membrane, and the beta receptor is a G protein coupled receptor. So it's going to activate adenylate cyclase, and adenylate cyclase is going to increase cyclic AMP levels, and cyclic AMP levels going to cyclic AMP is going to bind to regulatory subunit of protein kinase A and then activating protein kinase A enzyme. Once protein kinase A enzyme is activated, that is going to activate glycogen phosphorylase kinase, and the glycogen phosphorylase kinase enzyme further activates glycogen phosphorylase and keep that enzyme active. And the active glycogen phosphorylase, it is going to break glycogen into glucose 1-phosphate. This is what happens. Now on the other side, that's one way of regulating glycogen degradation in the skeletal muscle. Another way, whenever nerve impulse comes in, like action potential. Now the calcium will be released from sarcoplasmic reticulum in response to the action potential, and the calcium levels in the cytoplasm increases, and it will bind with calmodulin. So calcium calmodulin complex is going to activate glycogen phosphorylase kinase, and that further activates glycogen phosphorylase and increases glycogen degradation. And the third mechanism here is muscle contraction. When the muscle contraction happens, ATP will break into ADP, ADP further breaks into AMP, AMP level rises, and AMP acts as a positive allosteric modulator of glycogen phosphorylase, and glycogen degradation will increase. So like this, so exercise, nerve impulse, means epinephrine, retake, like this, epinephrine, nerve impulse, muscle contraction, all of them will regulate glycogen metabolism, especially on glycogen degradation. What about glycogen synthesis? Whenever a person is in restful condition after meals, when the blood glucose are sufficiently available, and glucose enters into the skeletal muscle through GLUT4 transporter, which is an insulin sensitive transporter expressed in response to the insulin over cell membrane. So the glucose gets in, and this glucose going into glycolysis, especially person is in fed condition now, resting, so glucose 6-phosphate kinds of builds up, and that glucose 6-phosphate is going to stimulate positively allosterically on glycogen synthase enzyme, and also insulin keeps glycogen synthase active, thereby glycogen synthase enzyme is going to convert the glucose 1-phosphate, thereby glycogen synthase enzyme, it is going to convert UDP glucose into glycogen, and glycogen stores are replenished. At the same time, when glucose 6-phosphate is elevated in the skeletal muscle under fed condition, it will have a negative effect on glycogen phosphorylase, and that's why glycogen phosphorylase activity will decrease. So that's how it will happen. So briefly let me write it down. So insulin is going to increase the activity of glycogen synthase on one side, and also insulin increases GLUT4, thereby GLUT4 will allow glucose in, and this glucose will be converted into UDP glucose. That is what is needed for glycogen synthesis. So on one side insulin is keeping glycogen synthase active by inducing it, and also glucose going into glucose 6-phosphate, and glucose 6-phosphate into glucose 1-phosphate and UDP glucose. So there will be relative elevation of glucose 6-phosphate, and that glucose 6-phosphate also has a positive effect on glycogen synthase, and it will have a negative effect on glycogen phosphorylase. So overall this UDP glucose will go towards a glycogen formation. So it is there here, glycogen is synthesized. So this is how insulin can increase glycogen synthesis under fed condition in the skeletal muscle, fed and resting condition, remember that. Coming to the overall summary of glycogen synthesis and glycogen degradation. So the table here is summarizing all the points that we have seen so far. Now first let's look at liver, and then we will go into muscle. So in the liver we are going to consider 3 conditions. One is under fasting condition, what happens in the liver; under well-fed state, especially carbohydrate meal, what happens in the liver; and under exercise and stressful situation, what will happen. All these points we have already seen with the figures and all that I have explained. So this is just an overall summary, very beautiful table here. If you can understand and remember all these points, that's what is necessary points for regulation of glycogen metabolism. Let's look at that. Now the fasting state, liver, and we are considering fasting condition. What will happen? So as you know, fasting condition means glucagon rises, insulin goes down, because increase in glucagon means increase in cyclic AMP, protein kinase A activated, that's going to phosphorylate glycogen phosphorylase, so glycogen degradation increases, and also protein kinase A is going to phosphorylate glycogen synthase, so glycogen synthase enzyme become inactive and glycogen synthesis decreases. Alright, so under fed condition, so when we take carbohydrate meals, what will happen? So glucagon levels will decrease, insulin level will increase. Since glucagon level decreases means cyclic AMP level decreases, so and glucose levels are high. So at that time what will happen? Glycogen degradation will go down and glycogen synthesis will increase. Now when a person is in a stressful situation or exercising, when there is an increase in epinephrine, and increase in epinephrine increases cyclic AMP by binding to the beta receptor, or it will increase calcium calmodulin levels by binding to alpha receptor. So overall what happens? Glycogen degradation will increase and glycogen synthesis will decrease, because glycogen phosphorylase is activated and glycogen synthase is inactivated. Now in the muscle what happens? Muscle fasting and resting condition. Let's consider that. Person is in fasting condition but resting. So under fasting and resting, so anyway there is no role of glucagon in the muscle glycogen metabolism. So we will have to look only at insulin and epinephrine's activity on the muscle. Now when the person is fasting and resting, so insulin levels are down, that means glucose transporters are not available sufficiently over the skeletal muscle membrane, so glucose is not coming in sufficiently, so glycogen synthesis will decrease and glucose transport under fasting and resting condition will also be decreased. Now, and also there is no need for glycogen degradation here, that's why glycogen degradation is not shown. Coming to carbohydrate meal when the person is resting. So carbohydrate meal and person resting, that's a second scenario here. So insulin increases because of the carbohydrate meal, And increase in insulin means glucose transport across the membrane using GLUT4 transporter is increased. And since glucose is coming in, so glycogen synthesis will also increase because glycogen synthase is kept active by insulin. Now the last scenario is exercise. When the person is exercising, epinephrine levels will increase. Epinephrine binding to beta receptor increases protein kinase A, and thereby it is going to increase cyclic AMP levels. And also by nerve impulse, calcium coming from sarcoplasmic reticulum binding to calmodulin, and calcium calmodulin levels increase, and exercise breaking ATP into ADP, ADP into AMP, AMP levels will increase. So all of them, they facilitate glycogen degradation, thereby glycogen degradation increases and they cause decrease in glycogen synthesis, and also there will be increase in glycolysis. As you see here, why glycolysis is increased? It is because AMP will have a positive effect on PFK1 enzyme, that is phosphofructokinase 1 enzyme. So like this, glycogen metabolism is regulated in our body by hormonally and also by allosterically. So I hope this video has helped you to go over all the details and all the points that are related with glycogen metabolism and its regulation. Thank you.